We at Regenexx Bio, our mission is to improve lives through the curative potential of our NAV platform of AAV vectors. And we've been leaders in the space of gene therapy across the space in terms of thinking of advancing the appropriate vectors, the appropriate routes of administration, how to administer these safely to allow for one-time durable protein expression for both monogenic rare disorders and also common disorders. And in fact, one of the significant milestones in the whole field of rare disease and gene therapy, of course, was Zolgensma for the treatment of spinal muscular atrophy, SMA, which is based on our NAV vector AAV9 to deliver that transgene. So that being the first ever approved systemic in vivo gene therapy for that devastating condition is a significant milestone. And we initially, as a company, in our early years, were focused on delivering our NAV platform of vectors like AAV9 and AAV8 to licensees who would advance them the way Novartis has, for example, with Zolgensma. But over the last 5 years or so, we've also focused on and really transitioned our priority to advancing programs ourselves. And we have 3 different platforms, one retinal targeted disease, where we deliver a validated target protein anti-VEGF to treat those diseases. But we also have other platforms, including CNS delivery to treat the significant CNS manifestations of certain lysosomal storage diseases like MPS II and MPS I that are currently in clinical development.
Great. And so we were just at WORLDSymposium, and we saw that you had data there on MPS I and II. Before we get into that, just an overview of these rare diseases. Usually MPSs seem to be broken out as separate diseases themselves, but I see you have data on both 1 and 2. So maybe just give us an overview of those 2 MPSs.
Sure. So as you mentioned, there are a range of different MPSs or mucopolysaccharidoses that are all lysosomal storage diseases. They all have in common, as other lysosomal storage diseases, a monogenic origin where the patients in each category are missing a particular enzyme that allows for the lysosomes to function properly and break down the various byproducts that exist within the cell. And in the case of both MPS I and MPS II, the respective enzymes that are missing lead to significant buildup of glycosaminoglycans, or what we call GAGs for short, and in particular heparan sulfate. And this is very key for several reasons. One is being monogenic disorders, and given our AAV vector platform, particularly AAV9, where we can deliver these genes via one-time gene therapy via these vectors, and again, particularly AAV9 in our approach, this allows to provide that missing gene so that those cells in the patient's own body can start producing that enzyme so that you can break down the glycosaminoglycans, and particularly the heparan sulfate. And the other benefit within MPS I and MPS II is that we know that these heparan sulfate buildups that occur throughout the body, and particularly in the CNS, are correlated with bad neurocognitive and neurofunctional decline or outcome for these children. And because of that, this allows for an opportunity to assess clinically with these biomarkers whether we're having an effect of our CNS delivered treatments. So we can do this by actually looking at the biomarker levels in the CSF. And we can evaluate as expected that these levels are very high in these patients before they receive the gene therapy. And we can evaluate for reduction in those, in addition to over the longer haul, evaluating the actual neurodevelopmental function of the children and also caregiver reported outcomes to over time correlate those biomarker responses to actual potential benefit for these children and their families. And so that's sort of the forward aspect of why MPS I and MPS II from a drug development standpoint we can go after, but why even evaluate this in the first place? And really that's what drives this, is that there's a really big unmet need for these children with MPS I and MPS II because of the fact that, yes, there are systemic enzyme replacement therapies that exist, but because enzymes given systemically do not cross the blood-brain barrier, what we're seeing is these systemic manifestations of these diseases are being better addressed by existing therapies. But the very important neurodevelopmental and overall CNS manifestations of these diseases are going on unabated for these children and their families. So this is really the outcry that we hear from our engagement with families and patient advocacy, and really speaking to the community that, yes, the enzyme replacement has been a great advance, but how can we help these kids to allow for them to develop from a CNS standpoint in terms of neurocognitive development, neurobehavioral aspects, skill acquisition, all these important things that are so important to these children and their development and the families. And that's where our AAV9 vector that we know has excellent tropism for CNS tissue, our leadership in developing route of administration where we can give our AAV9 vectors via local administration via intracisterna magna injection, which is an approach that we pioneered and advanced. We really have a unique opportunity to address this unmet need with an ideal vector approach and the specific gene. And we now, and this is why we're so excited about the recent results that were presented at World for both the MPS II program by Professor Giuliani and also MPS I by Ray Wang from CHOC, because we're being able to translate this potential into results that we're seeing in terms of some of the things we talked about in both programs. We're now able to, with each update, show longer and longer durability in terms of expression of the protein that we're delivering via one-time gene therapy, durability of the biomarker effect, actually reducing the key substrate, the heparan sulfate, in the CSF. And now, and this is key, now we're getting longer term follow-up with these patients where we can see the developmental curves, whether patients are indeed continuing to have meet neurodevelopmental milestones that we know they would not meet based on natural history data that we have with both of these conditions, even with, of course, systemic enzyme replacement therapy, where that is not really touching the CNS manifestations of disease. And another thing we like to point out is right from the beginning, we utilize the most validated approach for assessing neurocognitive development in the age groups that we're looking at. So that's the Bailey scales. And we felt it was very important to utilize that right from the beginning so that with the early patients treated in our trials, we can gather the most long-term data available to continue to make this link between the biomarker effects that we see and the neurocognitive assessments. In addition to that, this is the first time that we've shown additional neurodevelopmental functional data beyond the standard Bailey scales. And here we've incorporated caregiver reported outcomes, which of course are very important for these families. So they're both clinically meaningful, and they're also an opportunity for us to assess the totality of how one-time gene therapy may be benefiting children and their families. And we've seen really nice improvements on some of these measures, such as things like general behavior, maladaptive behavior, whether that is decreasing toileting, and also sleep. So, of course, hearing all these are all things that really matter to patients and families. So we're really glad that Dr. Giuliani and Dr. Wong were able to present this at the WORLDSymposium and to the LSD community in general. One thing I haven't mentioned, which I should have started with, is that we've seen excellent safety and tolerability, which obviously is of paramount importance. And I think that's something that we build on the learnings that have occurred in the field, and a lot of what's come from our own licensees, you know, that we have a lot of learnings across our different AAV serotype platforms and our different routes of administration that allow us to really design these different initial first-in-human studies that have a typical ascending dose cohort type approach. So now that we've seen excellent safety and tolerability, we're seeing what we want to see also in terms of dose response, in terms of the biomarker activity. We feel we're at the dose levels that make sense to be carrying forward in terms of seeing evidence of safety and tolerability and the biomarker effects and consistent functional advancement with those biomarker effects. The other thing we're actually seeing is, even though we're targeting CNS, we are actually seeing evidence of systemic spillover, where there's some evidence of enzyme levels, transgene levels systemically, and also biomarker effects systemically when we look at things like urinary GAG levels, which makes sense as evidence of systemic effect. So given all of those findings, that's what gives us confidence that in each of these indications, we've hit on the dose and the overall treatment approach for one-time therapy. So now we are expanding those cohorts in each study to look at more patients, to expand what we can learn about not only biomarkers, but also linking them to functional outcome in these patients. The other key benefit that we as a company have is our leadership in GMP production. And we know, like everyone else in the space, that a key rate limiter is supply of production, even for clinical trials, that this is turning into a real bottleneck for the industry. So we prospectively, several years ago, really prioritized having our own commercial GMP scalable production facilities in our own control, and not out of our control. In other words, with a CDMO or a manufacturing organization that we have to rely on. And that's very fortunate for us that we've done that. And we now, we announced, you know, that we do have our own internal scalable to commercial scale GMP production capacity internally. not only for scalability reasons, but also for quality, reproducibility, reliability that are so key in drug development for gene therapies. And I mention that because to your question of moving forward, we're not only moving forward with dosing more children, but in the case of MPS II with RGX-121, this expansion of patients is going to be at scale, so to speak, commercial scale, so that we can already be moving forward with the same product that we would intend for commercialization. Then I think from the key issue is alignment from a regulatory standpoint on what is a pivotal package that would be acceptable for these indications. And that again is why we're so excited about the recent WORLDSymposium updates, that it's really the maturation of this data and our learnings across the field that allows us to, with each data update, have more of a case in terms of why do we believe biomarkers matter, and why do we believe that those are reasonably likely to predict benefit for these children who have no benefit derivable currently in terms of the CNS manifestations, by also linking what we're seeing there with continued maturation of our functional outcome data. So our goal is to dose more patients, get longer term follow-up, and advance discussions with regulators on a package and a plan that we can align on for an eventual BLA for indications like these.